Animals are able to understand the world around them. We can all look at this image and quickly know what's going on in the scene. For example, I notice a picnic basket, this cheese, some apples, a cat, and an unusually large mouse. You've never seen this exact image before, and it doesn't matter if you see this image or smell the food or hear the cat. Our brains are able to quickly understand this scene and recognize all the objects in it. And our brains are able to do this because we are able to make generalizations across multiple versions of the same objects or categories. For example, if these apples are green or red, you still categorize them as apples. Cheese and apples are still categorized as food, where if you're the brain of this mouse, you better be able to quickly categorize the cat as a predator, or you're not going to be around much longer to eat this picnic. I'm really interested in how the brain accomplishes this. I'm really interested in understanding how networks of connected neurons in brain tissue represent information, and how this information results in our ability to perceive and recognize objects in the environment. I've chosen to study this question in the mouse olfactory system. Odors, the smell of natural odor objects such as cheese, are composed of complex mixtures of tens or hundreds of different chemical odorant molecules. We know a lot about how these odorants are detected in the nasal cavity of the nose, and how different types of neurons connect to each other and transfer this information across the olfactory system. It's known from human studies and using functional fMRI and psychophysics that particular areas of cortex contain the information for object identity and category. But how specific patterns of neural activity in awake behaving animals encode and perform this flexible categorization is largely unknown, and I'm really interested in trying to uncover some computations that are occurring there and the details of it. So to do that, I have developed a strategy to train mice to recognize novel odor mixtures, and then I record from neurons in their olfactory cortex both before and after I've trained them to recognize these new odors. I want to understand how the response properties of individual and populations of neurons in olfactory cortex perform these computations, and I hope to uncover, or at least potentially add to general principles that any system could be using to accomplish these tasks. And my name is Shtrahman, and I'm a postdoc in the Murthy Lab. Thanks.